All right. Welcome everybody. So this is our first real fall seminar of the semester. So to kick everything off, we have Mr. Shaun Donovan with the San Antonio River Authority with us today. And before before Shaun goes into his presentation, I'd like to welcome him, obviously, and give a brief introduction of Shaun. So Mr. Donovan is the manager of environmental sciences. Is taken directly off from the SARA's website, and he has been with the River Authority since 2012. And he's an aquatic biologist by training. He is an A&M graduate from Corpus Christi. I don't know what major. My guess is fish or biology in general. Yeah, and then
marine biology.
Marine biology. Yeah. And then he worked offshore for 2 years, doing research in the shrimping industry in red snappers and shark species. That's interesting. You might want to talk about that too. And Shaun is instrumental with the River Authority in developing and expanding ecological restoration efforts. And my guess is he's going to talk a few stories today. And he's a certified fisheries professional and active member of the American Fisheries Society that we just, talked about that conference in Michigan. And he's also a certified project manager professional. So he has a lot to offer. And with that, I'm going to give it to Shaun.
Yeah, appreciate it, sir. Thanks for letting me come and talk to y'all today. And I've slides set out, but I know we're not in person, so it's a little bit harder to do it. But if there are questions or comments or thoughts, or anybody wants me to elaborate on something, or whatever else it may be, please feel free to jump in and raise a hand, or just unmute yourself and ask something. I want to make sure that this is as beneficial for this group as it can be. And if there's other questions about the some of the specifics of our department, of what we do on a daily basis, I'm happy to answer them. Alex is one of the folks here in the department with me, and Alex can answer some questions as well if something comes up that I'm not as familiar with. But again, I want to make sure that this is as interactive as it can be. So please don't feel bad if you interrupt me and ask a question or a comment as we're going through, and then at the very end we'll have a chance for some questions as well. So I'm going to go ahead and share my screen. But again, please feel free to jump in and ask me any questions if you have any. So let me go. I think I'm going to get the right screen this time. Screen one. Are you guys seeing a background of them yourselves right now?
Yes, sir. Yes, we see the bridge.
Can you see like presenter mode there? Or is it a full screen?
Yes, sir. Perfect. We see your presenter mode.
Shaun, I'm seeing the presenter screen, so it's not a full screen that you probably want to share.
Is that better?
That's better. Yeah.
Okay.
There you go.
Good. All right. So again, the main thing we're talking about is monitoring and managing environmental health. And I'm really going to talk about kind of three parts of that is the monitoring, the identification of what maybe some source pollutants might be, or some causes of concern. And then ultimately, the reason why we do these kinds of things is to make on the ground management decisions, changes in the watershed, whether that's large projects, or if we're trying to curb pollutant loads or something along those lines. Like ultimately, that's what we're doing. What we're doing for is we're trying to find ways to affect on the ground change. And so that's ultimately what we're going to get to. But again, just a little bit of a background on what we do at the River Authority, and then what our department does specifically. And so whenever I say what the River Authority does, we have a 4-county jurisdiction. So it's Bear, Wilson, Karnes, and Goliad, going from the northwestern county down southeast to Goliad. But really our active involvement in the watershed, again, is watershed-wise. And so, or watershed-wide, excuse me. So we go all the way up to Medina County, Medina River. We go north to the Cibolo Creek in Comal County. And we go all the way down to Victoria and Refugio County, close to the coast. So this gives you a better idea of not necessarily our political jurisdiction, which I won't get into that part, but just really where our environmental sciences team is concerned. It is a basin-wide approach that we do. And just as a reference point, that red star in the middle there is where downtown San Antonio is. And the three things that you'll hear a lot of times associated with the River Authority are safe, clean, and enjoyable creeks and rivers. That's our mission statement. And one of the benefits of that is like a level of ambiguity. What is safe to somebody may not be safe. So I might think of like enjoyable is a good one. I might think enjoyable is a place that I can go fishing. Somebody else might think enjoyable is a place where you can go kayaking. Well, that ambiguity is really good. But in order to measure our performance, we want to make sure we're saying the same thing at the River Authority of what's safe, what is clean, and what is enjoyable. So safe for us at the River Authority is flooding. Obviously, you see here there's some serious flood issues. We're in flash flood alley, so we get a lot of really flashy flood events. So safety is a big part, and we have a lot of floodplain modeling and mapping. We do some dam operations and maintenance. We have predictive flood models, and so that's a big component of what the River Authority does. I'm not going to touch on this really much at all today. You also have the enjoyable part. Like I said, enjoyable is something different. Some people might not want to roller skate on the Mission Reach. Some people might, but other people might want to kayak. Some people might want to go biking along the trail. Some people might want to go fishing. So enjoyable can mean a lot of different things. Again, we define it as use of our park systems, use of the trail systems. But again, I'm not going to go into enjoyable much today. What I'm really going to dive into today is the clean goal. So the clean goal or clean objective is really what environmental sciences department is really associated with for the most part. Now, again, I'll do this caveat for the last time today is that there's overlap in all these things. And what makes the river clean can also make it enjoyable and can also make it safe. And the same can be true for the other two. But really what we're talking about when we talk about clean is water quality, the riparian health of the communities. So the trees along the edge of the riverbank. We're talking about fish communities, so biological communities, fish, freshwater mussels, aquatic insects, those kinds of things. Like that's what we're talking about whenever we say clean. And so again, the clean goal is largely associated with the environmental sciences department. That's the department again that Alex and I are both in. And we have this is not a completely comprehensive list of the positions that we have in our department, but it's pretty thorough. And so if you look at the top going down, our water quality scientists and our molecular biologists are our lab staff members. Now we have different career ladders. So like Alex is a water quality scientist 2. We have senior water quality scientists. We have water quality scientist 1, molecular biologist. Our data management specialists, those are pretty straightforward, as are the quality assurance specialists. We do have an environmental investigator. So right now it's only one individual. That person will do like environmental site assessments. Ronnie is his name. He'll respond to pollution events, or he'll be the first responder to like a fish kill or something. And then after that, after he does his initial investigation, he'll bring in the lab staff if we need samples, or the field staff if we need to go respond to a fish kill or some environmental concern out in the field. And then aquatic biologists and watershed ecologists, I think, are pretty self-explanatory as well. And we're going to talk about the stuff we talk about today are things that every one of these positions has a hand in doing. None of the projects or efforts that we do in our department are done by 1 or 2 people. They're done by the entire department, which is a benefit for a lot of different reasons. We'll kind of get into that in a little bit more detail. And then just a heads up too, on the right-hand side, this is one of our interns from a couple of years back, is we do have a pretty robust internship program. We had our first lab intern this last summer. We have at least 2 field interns every summer. And then sometimes we have what's called a Hutton Scholar, is a high school student that comes in and works with us. So, there are plenty of employment opportunities here over time. And if there's any interest in talking about these things in more detail, I'll be happy to follow up with anybody after the presentation to talk about some of these opportunities. And again, I have the screens up. So it's hard to, for some reason, can't see everybody right now. And if you have any questions, then please, like Dr. Dunn said, please put your hand up and I can come to you during the presentation. So the overview I want to go through today is, again, there's these 3 points I talked about earlier. Our monitoring models and source identification. And when I say models, I don't mean like we're plugging something into a computer and doing a computer model, but we have different monitoring models depending on what we need to do, what we want to accomplish. And 3 of these things, 3 things we really look at that are good case studies or examples are our Clean Rivers program, our bacterial source tracking efforts, and then using biological indicators to tell you something about the watershed. Then we're going to get into some analysis and visualization. And finally, we're going to finish off with some pollutant mitigation strategies. So the Clean Rivers program is something that, it's not something that I personally heard much about or really anything about before I started working at the River Authority. And really that's, it's a bit of a shame, but this is something that is done statewide. It's been around for about 30 years. And this really is the way that water quality is monitored throughout the state. So if you're going somewhere, whether it's in the Trinity River or the Red River, or pick your river, the Guadalupe, the Brazos, if you're going to a river system and they're telling you water quality data, they're probably talking about this program to collect the data. Again, this is operated by river authorities, but ultimately it is overseen by the TCEQ. And I'm not going to get too much in more detail with that, but ultimately all of this information goes up into the EPA, the Environmental Protection Agency, and they ultimately put out what's called a 303D list of impaired water bodies. So the EPA, after the Clean Water Act, created this oversight for water bodies throughout the country. They delegate that responsibility for deployment of these monitoring programs or implementation of these monitoring programs to almost every state. I think there's 2 that are not run by the state; they're run by the federal government. I think it's Massachusetts and New Mexico. But Texas's water quality program is run by TCEQ. So again, this is part of the Clean Water Act. The Clean Rivers program is what it's called here in Texas. But again, this is a hugely important thing to be aware of and to know about if you're interested in getting in the water quality monitoring world, because you will undoubtedly go through the Clean Rivers program in some form or fashion. There's also this link at the very bottom that I think is a really good thing for you guys to be aware of. It's called the Surface Water Quality Monitoring Manual, or SWQM, SWQM. There's 2 volumes. Volume 1 is kind of the Bible of how you sample water quality in the field, whether that's sampling it for metals or sampling it for nutrients or chlorophyll. That tells you how to collect water samples. Volume 2 tells you how to collect biological samples. So if you're going out and doing a fish survey, or if you're going out and doing, there's not much mussel stuff in there, but if you're doing a habitat survey, those are the 2 documents you want to go to. I highly recommend being aware of those. I know this is I think this is completely a graduate seminar, but as you're getting into the employment field here in Texas, if that's what your goal is, going to river authorities and being aware of that SWQM manual is really, really important. It's beneficial if you want to go in the lab world, or you want to go in the field world, or whatever area you want to go into. Being aware of these manuals is a really, really good first step. So just to give you an idea of the scope and scale of what the River Authority, the San Antonio River Authority does, and I'll just refer to us as the River Authority. It's just easier. This is our monitoring map throughout the basin. So the point of this is not to get into like this super heavy detail about each one of these different locations, just to show you again that geographical spread of what we do in our watershed. Some of the symbology that I wanted to point out is all of the blue triangles are what the River Authority does. We do have other river authorities that collect in our basin. So you can see in the northwestern top left part of the watershed, there's those the purple pentagons. I guess a little alliteration there. So that's with Bandera County River Authority and Groundwater District. In the very far right bottom corner, you can see there's a plus. That's the Guadalupe Blanco River Authority. But basically, I wanted to just show this part because it shows the collaboration we have with other river authorities, other regional entities. This is not just us. We do the majority of the sampling, but we do collaborate quite a bit with our partners. So whenever I say water quality monitoring, this is what I typically mean. And this list in the top left is not inclusive of all the things that we do. We typically collect what's called routine or conventional parameters. I think there's 12 of them. Most of them are up here. Something like metals is not considered a routine parameter, but it's something we collect pretty often. And if you look back at this map, what we do typically is sample each one of these sites every other month. There are some exceptions. Bandera County does theirs every quarter. The TCEQ field office does theirs every quarter, and GBRA does theirs every quarter. But for us, the stuff that we collect is every other month. So whenever I say we're going out and collecting every other month, for the vast majority of those sites, we're doing everything underneath that first sub-bullet, that water quality analytes. So nitrate, nitrite, total suspended solids, chloride, sulfate. We also do abiotic data. That's something our field staff does in the field. So we have water quality instruments that we put in the water. We measure dissolved oxygen, temperature, pH, and conductance. This gives us a good idea over time. And then we do field observations. For instance, what's the current weather like? If it rains, you're going to have water quality impacts. Are there people around? Is there a dog upstream that is defecating near the river? That might have an impact on your water quality observations. And so you want to take an idea of what's going on around you and then see what's happening. You want to record that. But this is what I say whenever I'm talking about water quality monitoring. This is typically what we see. Now, one of the big things we always ask ourselves, and is really important to think about whenever you're doing monitoring, whenever you're setting up a research project, whenever you're trying to answer some question in the scientific scientific forum, is why do we monitor? So the stuff I talked about a second ago is we monitor in that regard or that sense because we want to establish a baseline. We want to say, what are the conditions in the watershed? We want to know what they are. We want to do something repeatable. We want to do something, every other month where we have these specific locations so we can establish if we start seeing something go one direction to the other, if it starts getting much, much worse, then we know, hey, let's go figure out what's going on here. If it starts getting much, much better, hopefully that's a measure of some management measure we've had or some, watershed condition that's changed, and we can go look into that as well. Well, sometimes we have what's called intensive monitoring needs, like this example here. So we have a lot of paddling trails throughout the watershed, throughout the basin, primarily on the San Antonio River, but there are paddling trails in other water bodies. So what we want to do is we want to equip the citizens of the 4-county jurisdiction with information to know, is it safe to go out there and paddle, or is it safe to go out there and kayak? And so what we do, it's called current conditions for the river. So each one of these sites is collected every single week. So you can see obviously much more frequently than our other sample sites. But with this particular monitoring, we don't need to know what's going on with metals every week. We don't need to know what's going on with nitrate every week. We want to know what's going on with E. coli every week, because that's the closest kind of contributor to potential human health impacts. So what we do every single Wednesday is our staff goes out and picks up what's called a contract run, and it includes these locations. And as you can see on the far right-hand side, this E. coli bacteria result can tell people how clean is the water from a bacterial perspective on this Wednesday before I may go paddling on a Saturday. So you have the locations you're at, the time that sample was collected, and then what the E. coli result is for that day. And we have some information on these pages that can direct you to, like, okay, well, what does 88 mean? What does 300 mean? What does 66 mean? It gives people a good idea to go, okay, I feel comfortable going out and paddling in this area. So you can see, again, the point of this one was talking about if you have a different need other than routine monitoring, you can start doing what's called intensive or adaptive monitoring to really target something very specific. So to take that a step further, because this is the same type of monitoring, it's just more frequent, we can also do something that's called like bacterial source track is another good example of really diving into something in greater detail where the frequency may or may not be the same, but you're looking at really in-depth information. And so just a very basic high-level overview. Bacterial source tracking at its very raw form basically says that human gut bacteria is going to be different than cow's gut bacteria, than pig's gut bacteria, than, pick your animal, than a deer or a possum or a bird or whatever it may be. And so in order to go in and say, these are the sources of pollution we have in a water body, we can do bacterial source tracking and say 10% of this water body, the pollutants in this water body are from humans, 50% are from birds, and 40% is from just general wildlife. So that gives us, again, a really, really targeted thing. So not only are we saying, we know that there's a pollution issue here, now we're saying we have a really good idea of what the exact source of that pollution is. And the general idea in a watershed like San Antonio, and really just kind of general and watershed wide, statewide, regional wide, worldwide, is you can have greater agricultural impacts in rural watersheds or rural waterways, and you're going to have more of a human impact in urban waterways. And that human impact might not be actual human waste, but maybe it's like, dog. I mean, people aren't picking up their pet waste. Maybe there's like, more chickens in an urban area because people are keeping chickens in their backyard. So that's the general thought. And over time, we've kind of looked at like, well, maybe that's kind of not exactly true. And so we've started looking at over time and saying, well, actually, these urban waterways are kind of made up of the same pollutant sources as these rural waterways. And so, again, you can kind of really start diving in. But the important thing to note here, too, to kind of summarize this monitoring strategy part, is none of this is possible without that baseline monitoring. So we said, what's the problem here? Okay, there's a problem with bacteria, for instance. Okay, well, what is that problem? How frequent is that problem? So then we can do weekly testing. And then we say, okay, this is a pretty persistent problem with this thing through this baseline sampling, through this intensive sampling. Now let's really dive in and get really granular and say, the problem is consistent, the problem's in this part of the river, and the problem is cows. So how do we go and then try to maybe exclude certain feeding activities near a river? Or how do we put water sources away from the river so that this cow is not coming to get a drink of water in the river? They're going, half a mile away from the river to get a drink of water so that the system is naturally able to absorb the waste that comes from this animal. So again, you can kind of look at monitoring has different levels of purposes, different thresholds of details, but it's always important to ask, why are we doing this thing? So this is kind of that loop that we're looking at on a pretty routine basis. Is we always do routine monitoring. If necessary, we can go to this intensive monitoring idea and then to bacterial source tracking. That bottom one, you can kind of take that out and it could be any number of things. And so in this particular case, maybe it's bacterial source tracking. If we're looking at, maybe we're going to talk about biological monitoring indicators in a second here. If all of a sudden we're seeing issues with freshwater mussels, that intensive part might be for metals. That bacterial source tracking might be really diving into what type of metals, because copper really impacts freshwater mussels. And so this is not to say it always goes in this exact pattern as far as what that third step would be, but the routine monitoring to intensive monitoring to like very granular details is pretty consistent. The fourth part of this that we're going to talk about towards the end of the presentation is load reduction measures, or our load reduction measures. And so this, just to give you an idea of this example, is we have routine monitoring. On the right-hand side, you see an automated sampler. So this was some intensive monitoring we did in the Riverwalk. And what this was doing is looking at load reduction measures in this building behind these green doors. The San Antonio Zoo has a UV disinfectant system. So this is a load reduction measure. This is a UV disinfectant system, that as water comes out of the zoo, that water is being exposed to UV light. E. coli is dying off in theory and then coming out. So again, we did routine monitoring. We identified the problem as bacteria. One of the solutions is a UV system to treat this water. So kind of see what that looks like. Just to give an idea too, is the most time is almost always in routine monitoring. You want to be consistent again and again and again. You want to go out there and know what's going on. But the load reduction measures are almost always the most expensive. Because again, you're talking about construction, you're talking about moving earth, you're talking about building parking lots or building a UV system or installing a cistern or a rain garden. So while the time goes to the routine monitoring, ultimately it is very expensive typically to kind of treat or address some of these issues. And we're going to talk about that and kind of break that generalization down a little bit here, but just want to kind of talk about that. So before I get into any of this next, are there any questions where we are now? Because I know we're bombarding you with a lot of information. So are there any questions with the general idea of monitoring, why we monitor, what we're trying to accomplish, maybe other questions or thoughts that come up on that part of the presentation?
I have a question. I raised my hand too. My question is, for instance, the zoo, you say like urban monitoring. Can you hear me well? I'm sorry.
Yes, sir. Yep.
Okay. So the zoo puts out a lot of bacteria in the river, and how come we don't have them pre-treated first, or bacteria around the zoo? That's my question. I'm sorry.
Yeah, you're fine. You're fine. So for a number of years, starting early 2000s, they were still putting untreated water into the San Antonio River. And so that was that idea of, we had these long-term monitoring stations in Brackenridge Park near the zoo. We noticed, okay, why is Brackenridge Park, why is the E. coli concentration higher typically in the zoo than it may be elsewhere? So what we were able to kind of deduce from that was one of the sources might be the zoo, and Brackenridge Park in general, one of the sources might be the zoo. So let's do something where they are treating their water before it comes into the river. And so that's what that installation of that UV system is. So as water goes through this, it's kind of like a a canal system. It goes through these different paddocks in the zoo. As it goes through there, obviously there's animals living, it picks up bacteria and it starts to discharge into the river. Now that water is treated, and so that is a load reduction measure. And they've had really good success with that UV disinfectant system where I know some of this isn't in context, but the discharge numbers might have been 1,000 cells of bacteria per milliliter of water. Well, now they're about 1 or 2. So they have, over 99% reduction in bacteria coming out of the zoo going into the river. So that is one of the success stories. Is found a problem, identified a solution, implemented a solution, and now you have improved water quality in that area. And that's by no means the only problem with like Brackenridge Park, or for instance, or we haven't solved the basin-wide concerns here, but that is a good example of one solution that's been implemented.
Thank you for that.
Yes, sir. Yeah.
I think David has a question, right?
Yeah, just a pretty quick one. At the beginning, you were saying Sarah's motto is like clean, enjoyable, safe rivers. So I was just wondering, how often do you feel the need to sacrifice one of those for the benefit of another?
Yeah, that's a really good question, David. And so one example that I'll give a little bit more, I'll talk about a little bit later too, is like the Mission Reach, for instance. The Mission Reach is this huge restoration project that is part of a $385 million project overall. I think the Mission Reach itself was $270 to $290 million. That specifically, its primary focus has to be a flood conveyance channel. So that primary focus has to be safe. And so you may have to do things like a stem density removal, because if you get too many trees in the bank, you're providing too much resistance for that flood, for those flood waters, and it might start getting higher and higher than you expected it to. So in that case, we have to trim those trees back. It makes it less enjoyable because you have, fewer birds in the area. It also makes it, less clean because you're not able to filter that water as much coming in. And so there are certainly times where you have to go, what is the priority here? And so without, being able to quantify specifically how often that happens, it is a consideration that we have to make pretty regularly. So, I mean, that's a really good question and a really good point too.
Thank you.
Yeah, absolutely. Anybody else before I jump back into the biological indicators part? Awesome. Good questions. Thank you very much for those. Okay. I think you guys are seeing the presenter view again. Is that better?
No, we're still seeing
I'm
the
not
cover
seeing
page.
anything.
Yeah, just the cover page.
Gotta go back to this one then. That better?
It's the same on my screen.
I concur.
Let me try this one more time. How about that?
Yes.
There, it's back. Thank you.
Yeah. It's good because you guys are still, I can still see all right now too. So hopefully that sticks around. Okay. So the biological indicators are another really important thing to look at. And a lot of times, again, I use the word indicator because it can be an indicator of community ecological health, but can also be an indicator of water quality health, and vice versa. So this is a picture on the right-hand side of a health advisory sign that can be found at Leon Creek. On the left-hand side, obviously, is a fish. And so what is done in order to have this health advisory is we would do something like looking at the pollutant concentrations in the tissues of fish, because we want to make sure that we can, inform the general public to say, you can eat this fish or you should not eat this fish because there are maybe pollutants in that fish tissue. And what you can also do is you can look at the water quality. So say, for instance, in this case, the pollutant of concern here is PCB, so called polychlorinated biphenyls. It's an emerging contaminant. It's been around for a while. It's been in the public awareness for a while, I should say. So you can do two things. You can look at the water quality and you can look at the concentration in the water, but ultimately, if you're looking at it from a human perspective, which you shouldn't always do that, but if you are looking at it from a human perspective, you say, well, how would that potentially impact us? And one way that can impact us is bioaccumulation. So these like the food web, the medium fish eats the small fish, and the big fish eats the medium fish, and then we eat the big fish. And so This is an example of this bioaccumulation where there are toxic levels of PCBs in the fish tissue of fish from Leon Creek. So as you can see, there's a lot of things that go into that. Okay, there's obviously a water quality concern. There's obviously a fish tissue concern, and there's obviously a human health concern. And so that's a really good indicator that there is some larger thing going on in this watershed that we should probably look into, probably identify how do we go about looking at that. So fish and aquatic organisms can be hugely important in identifying sources of pollutants. In this case, this health advisory is just around what used to be Kelly Air Force Base. So you might have certain things that have PCB concentrations. PFAS, PFAS is something that's more and more in the public consciousness right now. That's a huge part of the Air Force Base thing. Like firefighter foams are really common sources. And so you're seeing a lot of these different emerging contaminants show up around military bases or show up around industrialized areas. And so again, biological indicators can be really important for telling the health of a system. So we do this in a number of ways. The kind of went a little bit of a reverse thing there where that fish tissue part is that's the intensive part you would do later. That's like the equivalent of the bacterial source tracking. It takes a lot of time, it takes a lot of money, really, really important, but it's not something you would just do as a recurring means or method. These are things that are very much recurring methods that we have. The index of biotic integrity is something that's created by the state. It basically looks at the entirety of a fish community and talks about what is the health of that fish community. You can learn a lot by what specific fish are there. So the top right, everybody probably recognizes that as some sort of a catfish. Catfish are typically very tolerant species. They don't mind big fluctuations in water quality, big fluctuations in temperature. They can have really low dissolved oxygen tolerances. Whereas the fish in the bottom there is much less tolerant. It's called a gray red horse. This is something that is a really good indicator of a healthy system, a really good indicator of maybe a recovering system. And so again, different species can tell you quite a bit. So we also have intensive fish surveys. We do one every year or every other year now in the Mission Reach. It's called our intensive fish survey out there. And that'll tell you a lot of things about habitat associations, what kind of areas do they like using. And so again, you can learn a lot about the biological indicators, again, being fish. You can also look at it in a pre or post restoration monitoring. So stream restoration is a solution method, a mitigation strategy I want to talk about in a couple of minutes. And you can use these at the tail end of that restoration cycle to say, this restoration is working, it's being successful, or it's not because of these indicators. And so again, tells you a lot. One thing that people aren't as familiar with that is really, really important are these called freshwater mussels. So those first two things contribute to the kind of the phrase here in the third is freshwater mussels are filter feeders. So they filter water through the system. They extract what they need, what foodstuffs. They can eat bacteria, they can eat algae, they can eat different, very small floating particles in the water, different detritus in the water system. And that's why a lot of times they're considered kind of the liver of the river. And to kind of run with this analogy, maybe, we'll see how it goes, is as people know, the more toxins you take into your body, you might be damaging your liver. And it's the same thing. If there are toxins in the river and these mussels are filtering them out, they're going to accumulate those toxins and might be an indicator of there being a problem. And so an example that I'll give is when I was up in, I got a chance to go to West Virginia for a conference, and there's a river there that's beautiful. It's really clear water. All of a sudden, there was nothing physically, visibly different about it. All these mussels started showing up dead, and all these shells ended up being opened. These mussels were dying, and they weren't sure what was happening. There were, again, no visible changes, no big land use changes. So they took some time and figured it out. They ended up finding that this company who was applying pesticides, that the state was doing this pesticide application. They were using a new formulation of this pesticide application for black flies up there in West Virginia. And it turned out that was extremely toxic to these mussels. And so you never would have known it if you were doing your routine water quality sampling. You never would have known it from odor or sight or anything. But these mussels started accumulating these pollutants in their system, and they ended up dying off. And that's what told people, hey, something's going on here. So this is a really, really important species. We're doing a lot of work right now to do a freshwater mussel reintroduction into the Mission Reach, the restored stretch I'll talk about in a second. But again, mussels are really, really important. They're really good bioindicators. And unfortunately, they're very susceptible to things like ammonia and copper. Those are the two things that they're the most susceptible to. But again, these, just by observing these populations alone, you can get a good idea of what's happening with the pollutants in a river system. The last thing that I'll talk about a little bit on the biological indicators and the monitoring in general is this eDNA as a factor that we can utilize. So eDNA is called environmental DNA. The animals like slough off this DNA into the water system. So without actually having to find the animal, you can filter water and say, we know that there's zebra mussel DNA here, which even though we haven't found the zebra mussel, that gives us a pretty good indication that this zebra mussel is in this water body. We do eDNA monitoring right now for zebra mussels in our lab. We do a few samples in San Antonio River and Medina River and Calaveras and Browning lakes. Those are all zebra mussel negative lakes right now, so they're not infested. Medina Lake in our system is infested, so that's why we're very, very closely monitoring to see if this species starts spreading. So I won't go into much detail on this one, but just another good indicator of ways you can use monitoring and mitigation strategies to kind of impact these things. Okay, so I'm going to go into data analysis and visualization, and I'm going to get out of this presentation just for a couple minutes, and I want to show you guys. I want to show you guys some visualization products that are available because these products are available to anybody. These are external sources that people can go and take a look at. There is a link on the slide, and I pull it up in a second here. But I just want to kind of show you guys how you can utilize these tools, what you could potentially do with them, and how they're possibly helpful for y'all. So, are you guys seeing people pulling a seine? Are you seeing the front page of our website right now?
Yes, sir. We're seeing it.
Yes, sir.
So on this page, this is our homepage, the River Authority website. So on the top right, I think this is called a hamburger, I think is what they call it. You can click on this little part, this little stacked area on the top of the web page, and there's right here in this bold letter, it says maps and data. If you go to maps and data, there's these 6 agency interactive viewers that we have. The top left is floodplain viewer, risk map. The two that I'm going to really talk about for a second here is water quality viewer and our bacteria dashboard. But again, we have some viewers here. If you ever need water quality data or information for a project, or you're curious and doing something for a thesis study, all this information, everything we collect is publicly available, and you guys can use this if you're interested. So we're going to go to the water quality viewer, and I'm just going to walk you around just for a minute here. So what you'll see at the top is these different tabs. So water quality, primary contact, recreation, aquatic life use. We're going to focus on water quality. So the really cool thing about this is it shows all of our stations that are current, historic, or recreation sites. Our recreation sites are the one I talked to you guys about that we do every single week for paddling purposes. So I'm going to pull up one of those. And this is one of our sites at the very end of the mission reach. And this is called Loop 410. So we can click on this site, or we can go to station information. And then it's going to start getting a little bit more granular for you. So what it's going to show you is the E. coli count at the last sample. If we've done bacterial source tracking in a county, it tells you this is the percentage of human bacterial cells and pets and cattle and non-avian wildlife. So you can start getting really granular in this information. You can look at things like aquatic community. So if you go to this next tab right here, it'll tell you, how's the dissolved oxygen? It'll tell you what the nutrients are like, and tell you what nutrients, what do we mean by nutrients? So it'll tell you nitrate, total phosphorus. So as you can kind of see, again, we have all the data that we collect, but if we don't analyze it, if we don't visualize it in some form or fashion, then we're not going to be able to see any trends. We're not going to be able to identify anything as issues. This is really, really important part of what we do at the River Authority, and really what monitoring is about, is being able to identify these trends and these changes over time. So you can look at, again, all of these 4 tabs. We try to make them as digestible as possible. So total suspended solids, total dissolved solids. As you've seen, some of these have green bars. Some of these have red bars. So green, as with usual, means good. Red means bad. So this nitrate level is pretty high. Well, that's common in our basin. We understand that. Bacteria level you saw was 6. That's really good. The magic number is kind of 126. But what you can also do is hover over some of these things and see what has been in the last 12 months. You can see all the data points that we have over time. And this really allows us to step back and say, hey, what's going on with this? What are we seeing? How do we analyze it? We also have some internal viewers just to kind of show you what we do with some of the internal analyses that we do, ways we look at data. So we have a lot of these different viewers. Really the one that I kind of want to go into just a little bit more detail is this bacteria source tracking viewer. Talked about that as being really important to dive into what's going on. But again, data analysis, huge part of what we do. So we could come into this site or this page, and we could say, what have we done in 2023? It'll break it down to these number of different events we have and these different spots. So we can click on that and say, for instance, we talked about Brackenridge Park earlier. So if we went up here to Brackenridge Park and we clicked on, let's see, actually get out of that. So if we came over here to Brackenridge Park and we said, what's going on at this site, we can get a really good idea of the concern of Brackenridge Park is usually non-avian wildlife. So that means things like deer or possums or foxes. But if we go further up in Brackenridge Park, then we can start looking, actually avian wildlife jumped up a lot there. So maybe there's a bird problem. Again, it's just a really good example of making sure that you look at your data. And I think the interesting part about this too is you're not necessarily trying to target what you're looking for all the time with data analysis. Sometimes it's important to do that, but sometimes you want to visualize what you have or analyze what you have from a broad scale and see what comes out at you as maybe a point of interest or a note that you have from looking at something. So again, I just wanted to share those data analysis parts with you guys for a second, just so you can kind of see. And then the last thing that I want to talk about here in the last couple of minutes is looking at management, pollutant mitigation strategies or management strategies that we may or may not have available at our disposal. So let me try sharing my screen one more time and we'll wrap up here. Everybody see that okay?
Yes.
Yes, sir.
So there's really two ways to look at these mitigation strategies. It's we kind of break them down as planning and doing. I'm not going to talk about the planning a lot. The doing is the bigger part I want to focus on today, but there are a lot of plans out there. And in my moment of cynicism, a lot of these are things that do sit on shelves and gather dust. Watershed protection plans are things that people can volunteer to be a part of. So our implementation plans, total maximum daily loads is you can do a calculation to say how much of a pollutant can go into a stream at any given day, with that stream still meeting any sort of water quality standards that are set for it. So again, these are plans, these are metrics, these are calculations that can go into the potential doing part. But again, there a lot of times, unfortunately, those do end up sitting on shelves. And so I want to talk about some of the doing items that the River Authority is involved in. So whenever you look at like what the problem is a lot of times you see this as a model for us. A manicured surface might be a park with grass all along the edge of the river, mowed all the way down to the edge, or an impervious surface like a roof or a street. And you have the water flowing down or falling down, like rain, for instance, and you have it going over these surfaces. So over a plain surface, on your manicured surface, you could have all these different pollutants. A lot of things we talked about today. On your impervious surface, you can have those plus other ones. Heat is a big one. Obviously, urban heat island effect, climate change things we're seeing. You can have a lot of these things coming into the river. And so one of the big strategies that you have is how do you reduce this amount of plane flow over the plane surfaces, a sheet flow? How do you slow that water down, spread that water out, and let the system naturally, kind of take hold in things? And so this is very traditional stormwater management. So you can see on the left-hand side, working over to your right, you have, maybe a big box store, and you have water falling on the roof, getting shot out into the parking lot. That parking lot eventually drains into some water body, maybe that drains into this channelized ditch first, and it eventually makes its way into a river. Well, that water is not doing any slowing down. It's not doing any spreading out. It's not doing any natural filtration. It's just going over these impervious surfaces right into a system. So one of the big things that we're trying to advocate for are these litter BMP features. So low impact development or best management practice. You can kind of see an example here where there's these curb cutouts. So in theory, if this was rain in the parking lot, it would run off into this rain garden. This rain garden would be able to absorb, things would go through the plants, things would go through the soil, it would come out this under drain, and it would eventually make its way to a storm area. So for an example, this is just outside of my window here. This is a rain garden that we used to have. Water would come off the roof, go into the rain garden, get absorbed into those soils. Pollutants would have a chance to go through natural biological processes and reduce the amount of pollutants going out. And so some real world examples of other things you might see are like a cistern. A cistern doesn't necessarily spread it out, but it slows it down because you're capturing that water. You're not letting it run straight out. And then as you can see on the pipe here on the left-hand side, this drains out from the top. So what happens is all these pollutants settle down at the bottom. And as it overflows, it goes out of the top. You can eventually open this up and drain it out. You can drain it right here into this grassy area to let it slow down. You can drain it into a truck that you're taking away to take somewhere else to a treatment facility or something. Another cool example is what's called permeable pavers. And so this is traditional, asphalt parking lot. This is a permeable surface. Water can filter into this parking lot. And then some examples, like we talked about those curb cutouts that we saw in that one animated image. Here's curb cutouts going over this rocky area right into a rain garden. This is at the Duzam. And you can also see this is Broadway, and there's a little curb cutout here. So as opposed to this being a storm drain, this water from the street will run off into this little channelized area. It'll get a chance to settle out here where these trees and plants are, and that water can get filtered out as well. Another really good thing that is admittedly a little bit of a pricier option is stream restoration. So this is the Mission Reach area south of downtown. This is what it looked like 25 years ago. You see it is grassy, but it is a channelized, trapezoidal channel meant to push water out. Over time, we basically regraded these banks. We put more bins into the system, and this is what it looked like during construction, and this is what it looks like post-construction. So you can see a lot healthier bank system, a lot more trees and shrubs and grasses. If water falls on this bridge, it gets filtered down through these trees. If it falls onto this walking, hiking trail, it goes through the grasses and the trees and then into the system. So it's much more of a robust filtration system. So those are two really good examples. Again, lid BMP features and stream restoration. The one thing that I admittedly, haven't known enough about over my career, and certainly didn't know enough about it in my education, is policy. I think people hear policy a lot of times, they think, oh, that's boring or not terribly interesting for me personally. But policy really is one of the biggest impacts you can have from a water quality perspective. So we have two things here in San Antonio called, one is the Unified Development Code. That's not something that's just in San Antonio. A lot of big cities have Unified Development Codes, but basically, as you see there in italics, it establishes standards and procedures for new development in the city. The reason why this is really, really important is that if you say, you can build whatever the heck you want to, wherever you want to, somebody could put a ton of impervious surface as close to the river as they wanted to. Whereas if you have Unified Development Code and these codes are telling you, you can't build certain things in certain places because you want to protect water quality, or you want to protect, stream health, you want to protect this bird population or fish population, you can really make a big difference. And two examples of that are what are called Rio districts. And there's Rio 1 through 6 from the San Antonio River. Rio 7 is a new San Pedro Creek overlay. The Rio 7 was just created in 2016. And to give you an example of what you can actually, what this looks like in practice in the real world is, let me see if I can pull up. Let me know when you guys see Google Earth. And I know probably too much screen sharing today because it's kind of messing me up, but let's try getting here. Can everybody see the Google Earth overlay?
Yes, sir.
Okay. So I'm going to give you a real world example that we were talking about yesterday with SAISD. So this is San Pedro Creek. Again, this is where the Rio 7 Unified Development Code district is. What this Rio 7 says is that you cannot build new impervious surface within a certain distance of the creek. So this area right here is a parking lot that is owned by SAISD. Excuse me, it's actually owned by the city of San Antonio. SAISD is a current user of that property. And what the conversations about this have been is, let's make this a parking lot. Well, because it's in this Rio 7 district, it cannot become impervious surface because it's new and it's a certain distance from the creek. So right now, while it's not a lush riparian ecosystem, it is a permeable surface. It is grass, it is rock, it is dirt. Stuff can soak into the ground there as opposed to running straight off into the river. So this is a very real example of right now, the only reason this is not a paved parking lot is because of this Rio 7 district overlay. So again, policy isn't always the funnest stuff to learn about or the most important thing whenever you look at it, at the very first glance. But policy is hugely important, especially in a town like San Antonio that is very, very big and there's constantly development going on. And so I would advocate for, if you have an opportunity to learn about policy or to know more about that, It's a hugely beneficial thing because again, it has a tremendous impact. It can have a tremendous impact in a positive sense, but it also have a tremendous negative impact. So with that, I know, been about 45 minutes or so here. So thank you very much for y'all's time and attention. I'm happy to take any other questions you may have if there's time for that. But again, thank you very much for letting me come and talk to y'all today.
Well, thank you very much, Shaun. So the floor is open. So we have about 10 minutes left. So any questions are welcome.
I have a question real quick. Does Sarah do a lot in the way of treatment, or is it more so like restoration and mitigation? So whenever y'all get like really bad results, y'all do treatment, or y'all rely on other agencies to take care of that?
We do a lot of remediation, and we do have treatment facilities. And so, that stuff, before it hits the river is really the best place to treat it. It is really difficult once it's in the system to do in-stream treatment. We are looking more and more at doing things like called tertiary treatment systems. And so you might have something where once it's in the river, the pollutants are hard to get out, but maybe if you do additional wetland type vegetation along a river or a creek, and you're able to divert some of that water through a wetland, then you have a natural kind of in-system treatment system or in-system treatment train. That is a really helpful thing to have. We do things like remediation for pollution events. And so we might have, if we have a pollution event along the river, we might have somebody go out and we'll remove those contaminated soils or remove those contaminated materials along a riverbank. We do things like putting in stream booms. If there's something like a petroleum spill, we can put in booms at the top of the water body to help filter out any sort of gas that's in the system. So we do some treatment, we do some in-stream removal, but a lot of the things are a lot of the methods we have are trying to get that water before it hits the river system.
I also have a question. Sorry, I'm having trouble with my hand going up and down.
Good.
You said you're monitoring the river for PCP, and I'm wondering why. Are you planning on? Are you trying to pinpoint where it's coming from? Are you planning on? Or you just want to monitor it?
So we're monitoring for called PCBs, polychlorinated biphenyls, and so PCBs. The reason why we look at those that family of compounds is because it's something that accumulates in fish tissue, and it's something that is harmful to humans if you ingest them. So that's why we're looking at that. That's long been kind of a known pollutant in the system. And so again, there are established methods for that. We don't do that analysis in-house. We do some of the fish collections and send that off to another lab. But yeah, we do stuff for PCBs, B as in boy, kind of analysis that we do.
But in addition to that, are you able to say, okay, it's more present here than it is upriver? And so you're able to say, okay, well, we see it's coming from this part of the river. Are you trying to focus in on that, or are you just trying to monitor for the fish?
Yeah, we definitely want to. So that's another good follow-up question is right now, the only part of the river basin that has a fish consumption advisory. So basically do not eat these fish is Leon Creek, close to those military bases. Every other place that we've done fish tissue analysis is good. And so that then tells us, okay, how do we remediate these types of chemicals in in that area, that part of the watershed? The good thing about that is that a lot of the activities that would have led to that introduction of those things are done. Kelly is no longer an active military base. It's Joint Base San Antonio. They do have an environmental wing within Joint Base San Antonio to do some mitigation work. But certainly if we found something that was concentrated in one part of our basin, it absolutely is kind of a red flag to say, let's follow up on this. Let's learn more about this particular area. Are there mitigation strategies we can implement based on this localized pollution? So yeah, that would absolutely be an example of like a red flag for us to really focus on in an area.
Thank you for that.
Yeah, absolutely.
Crystal, far away.
So it looks like y'all do a lot of monitoring, of course. I was wondering, have y'all tried like new technology to monitor some of these areas? I'm only asking because I'm doing an undergrad research and mine's using like drones with multispectral cameras in order to like monitor something like phosphorus and nitrogen to get some of those values that y'all may see in y'all's water quality data that you show.
Yeah, that's a really good question, Crystal. And we do try to explore new technologies for ways to look at water quality data. One of the ways we do is similar to that. It's not using a drone, but it is using a camera to look at, trying to look at concentrations of different constituents that USGS is doing. So we have a contract with USGS that we do routine gauge monitoring. So if you ever looked at flows in the San Antonio River on the USGS site, we're supportive of those efforts. They're looking at utilizing cameras to see, can we look at concentrations of pollutants on a real-time basis without having to do water samples. And so those are things we explore. We don't develop them ourselves, but we do get approached by a lot of vendors or third parties that says, hey, we want to try this technology in your basin. We try to sample, or we tried an instrument a couple of years ago, maybe 3 years ago, that their claim was that they could look at E. coli in real time using a water quality probe. So right now it takes about 18 or 24 hours to bring our samples back to the lab. Lab analyzes the E. coli results and we get them the next day. They were claiming they could do something real time. You could go look on the website and say E. coli values are this right now. And we tested it and we didn't have results that we thought we were confident in, but we're always open to testing new things because like efficiencies and streamlining what we do is a hugely important thing for us, because then it allows us to let our resources go farther and farther if we do find something else. So, certainly if there's something that you're coming up with or something interested in looking at, we're always open to discussing collaboration opportunities with universities or with different agencies. So, I mean, anything that you might think of as a unique or novel idea that might be worth giving a shot, then we'd love to hear that kind of stuff.
Perfect. Thank you.
Yeah, thank you.
Alex?
All right, I got two.
All right.
First off, how does the River Authority incentivize lid features or BMPs? And the second one is, to your knowledge, do the Rio districts have water quality standards set for them, or are they mainly construction standards?
So I'll do the second one first. There are not water quality standards specifically associated with the Rio. It is for construction. Obviously the goal and the idea is that we would be supporting the achievement of the already set TCQ stream standards. So there's not like discharge requirements. It's basically enacting methodologies and systems that we believe are in place, if in place, would have positive impacts to water quality, or certainly not detrimental impacts to water quality. The incentivized thing is certainly tougher. As you know, Alex, we had the watershed. We used to have something called a watershed wise rebate program where we would do different grants with different schools. We typically did high school, middle school, elementary schools, where people could apply, and then we could give up to a $50,000 award for them to put a lid feature on their campus. And so some of the pictures we have are things like Floresville Middle School or Floresville Elementary School or Warren High School here in San Antonio had these lid features on them that we incentivized. We didn't get a large ground support from that, and so we've actually cut that program. We're trying to do now more nature-based solutions, green infrastructure things ourselves on properties that we maintain and monitor, because we can have a greater confidence that those things will happen long term. But the incentivizing thing is a huge conversation piece and ultimately a goal, because if you can't incentivize the development community, or you can't incentivize people from a monetary, development and from a monetary perspective, or people from a monetary perspective, you're not going to get traction long term, because the truth is not everybody can make environmentally conscious selections with their money. Typically if you have the more expensive houses or the more expensive apartment complexes are going to be the ones that have more space and all this landscaping, where the vast majority of people make their selections on home based on affordability. It's not about all these amenities I have. And so how do you make it so that these developers can put these practices that are sometimes more expensive into a community while still making sure it's affordable for the vast majority of people living there? So those are, hugely important questions that, I certainly don't have the answers to, and the River Authority is trying to work with the proper groups to see like what could we do, how do we incentivize this more and more. So yeah, I mean, good question, Alex, and whenever you get the answer, we'll you can be implementing those for us as soon as you can.
There you go.
Hunter?
I have one question too. It's about endangered species. Does Sarah have a program to where they monitor stuff like the monarchs or like milkweed or different types of plants in the river that are endangered? Texas, is it Texas? I forget the endangered species in our river, but
we do. Yeah, we do quite a bit of endangered species work. A lot of it's collaborative with folks. There are not a lot of aquatic endangered species in the river system itself. So, milkweed is something that is a terrestrial plant that we do a lot of plantings with. We incorporate milkweed a lot of times into our operations and maintenance areas. There are some, endangered plant species close by, like Texas wild rice is more like a San Marcos species. Those are typically like spring associated, and so they're part of the Edwards Aquifer Habitat Conservation Plan. But again, most of those things are in San Marcos, a little bit New Braunfels area. Really, the biggest aquatic ones are, again, aquifer species. So you have like blind catfish or blind salamanders that are typically found in the aquifer. They say that they exist in our basin, but only because they exist in our geographical overlay of the basin, not on any surface water for us. And so we had a candidate species, a candidate freshwater mussel species a number of years ago that's no longer on the candidate for the Endangered Species Act. But we still do a monitoring for all these species. Should something happen, we have information on hand to be able to then share with the appropriate authorities if there are listing decisions to come down the road.
All right, I think Hunter has a question. I think that will be the last one.
Great. Earlier you brought up a term, I think it was called load management, connected with UV disinfection. Is that right?
Yep.
I was just wondering, since that's the most expensive cost, like what's the typical for the River Authority, like the typical steps to do that or the process of it? Because I imagine it's not as simple with how much more expensive it is.
Right. Yeah, it's a really good question too, and it's all about scope and scale. So for instance, one of the things I showed was like those permeable surfaces in a parking lot. That's our parking lot over at our main headquarters. So in that case, we own the property. We have a desire to do this. Let's go do it. And it basically is just, we have an elected board of officials. They approve our budget every year. Our executive staff and our staff will go and say, we want to do, $100,000 for a new parking lot and a cistern over at Gunther, get approval there, and we implement. The complete other end of the spectrum would be something like the Mission Reach restoration or the pending West Side Creeks restoration, where the West Side Creeks, we have $150 million of federal funding. And that took over a decade to advocate and work with the Army Corps of Engineers and work with locally elected leaders and state senators and people throughout the local, state, and federal governments to get those dollars approved. And so it can be anywhere between those things. We do a lot of projects with City of San Antonio, a lot of projects with Bexar County, where there is a lot of political part to it, but it's obviously local, so it's much less involved. So again, it can range all over the place on what some of those load management, load reduction strategies could be, from the wildly simplistic to the wildly over complex. Like the West Side Creeks money was actually something that was earmarked in President Biden's budget this last year for $115 million. We were the only project nationally to be earmarked that specific way by the Biden administration through the Army Corps of Engineers. And so that was through 10 or 12 years of advocacy. So it can get complicated. As another plug for like the policy political side, because a lot of this does require that long-term, consistent political will and foresight and thought going into it. And so again, I'll be the first one to admit that whenever I was going to school, when I was early in my career, I was like, I don't care about any of that stuff. I want to go out and I want to catch fish and I want to go on a kayaking trip and I want to go on a mussel survey. And like that stuff was fun and was important. But at the end of the day, that stuff, you combine that with the policy, with the political kind of side, like that's the sweet spot to ultimately be at. So if you guys are taking policy classes, those are probably good ones to make sure you're tuning in for.
Thank you.
All right, awesome. So with that, I'm concluding today's seminar. And I guess one thing that we couldn't do very well as an online program is we can't really give you a huge round of applause. But before you leave, say thank you to show our appreciation. All right, thank you.
Yeah,
Thank you, Shaun.
I appreciate
Yes,
everybody.
thank
Thank you
you.
very much for your time today.
Thank you.
Thank you.
Thank you, Shaun.
Appreciate it, guys.
All right, bye-bye.